Okay, so hi, thanks for being here. So early in the morning. And I am Marco, I'm a postdoc in Babak Momeni's lab, third floor. And our lab delves into the dynamics of microbial population. We have a part that is dedicated to the mathematical modeling of the bacterial dynamics and a part of experimental biology. Nicole came, as you all know, a couple of months ago to work into our lab, and I've been showing her the ropes of most of the experimental work we've been doing on a very preliminary project in its preliminary phase, which she will shortly be talking about. And so, in these two months, it's my opinion and Babak's opinion as well that Nicole has shown great dedication, motivation, intelligence, and curiosity. I mean, any three of these four traits are more than enough to make a fine researcher in the future, so good start. And also she has been working on this project half of the time by herself, because I had to leave for 3 weeks. After 2 weeks I showed her the ropes, so she actually handled herself pretty well. Made me realize how unnecessary I am, of course. At the same time, she grinded data, and that was a real help. So she's a student, an undergraduate student at the University of Miami in biomedical engineering, and she really did a nice job. So go talk about it.
Thanks. Good morning, everyone. Thanks for coming. I know it wasn't early morning for some of us. So my name is Nicole Thornton, and as Marco said, I am a biomedical engineering student at the University of Miami. I'm here as a part of the REU program, and I've been working in Professor Momeni's lab for the summer. So this summer, I worked on using artificial selection of bacterial strains to degrade mycotoxins. So mycotoxins are highly toxic fungal secondary metabolites. They are carcinogenic, teratogenic, hepatotoxic, and mutagenic. They are found in many different food sources, mainly corn, wheat, sugar, and milk. And the Food and Agricultural Organization actually reported the presence of mycotoxins in 25% of the world's food crop. And the toxins can contaminate foods during processing, storage, or transport when conditions are favorable for mold growth. So I have just an example of how toxins might enter into the human body. So crops such as corn are harvested and stored in silos where the humidity and temperature are conducive to fungi and mold growth. Here the fungi grow and they produce mycotoxins as secondary metabolites. The food are then ingested by animals and people directly. In addition, as toxins pass through cows to their milk, the toxins are magnified and transformed and then drank by people. And as the toxins accumulate in high concentrations in the body, it can be very harmful and even deadly. So our toxin of focus for this project is aflatoxin B1, or AFB1, which is a prominent mycotoxin that disturbs protein synthesis by transcription inhibition. It is produced primarily by the fungi Aspergillus flavus and Aspergillus parasiticus, and it is fluorescent due to its aromatic ring that you can see here. And this will be important to remember later on as I go through the methods and experiments. So AFB1 is the most potent aflatoxin, and it is the most toxic naturally occurring carcinogen known. It is known primarily as a liver carcinogen. And acute exposure are most likely to occur in developing countries where food handling and storage processes are suboptimal, where malnutrition is widespread, and where few regulations exist to protect exposed populations. And approximately $500 million in the US and over $1 billion globally are lost annually as a result of aflatoxin toxicity. And these costs are especially detrimental to developing countries' economies and food supplies, as they can't handle the burden of that. So current management of aflatoxins includes prevention, regulation, and detoxification, but chemical and physical methods can be extremely harmful to the environment and human health, so the scientific community strongly advocates for bioremediation or using microorganisms to break down environmental pollutants. So there are 5 things that we consider when we're considering what makes a good bioremediator. So that they are fast and efficient at degrading toxins, that they produce safe reaction products, that they are non-pathogenic, that they are applicable in real-life scenarios, and that they are applicable to the broadest spectrum of toxins. So based on these traits, we elected to use the genus Rhodococcus. And these are aerobic gram-positive bacteria, and they're considered great degraders due to their metabolic diversity and their wide catabolic potential. And that's associated with their large genome and their linear and/or circular plasmids that they may have. Rhodococcus bacteria are believed to play a critical role in the removal of polyaromatic pollutants from the environment. And Rhodococcus erythropolis specifically has found to be the best AFB1 degrader of its genus, and so the toxin that we focus on mainly in our experiments. So our ultimate goal is to find suitable biological detoxification procedures to safely remove mycotoxins and improve food quality globally, starting off with some smaller aims. So first we wanted to identify at what toxin concentration AFB1 begins to have an antibiotic effect on the bacteria. And then after that we wanted to employ artificial selection to improve those degradation dynamics. So throughout the experiment, we use a specific strain of Rhodococcus erythropolis. We use AFB1 and acetonitrile. We use non-fluorescent culture medium, both buffered to a pH of 7.2 and unbuffered. And we use a Synergy microplate reader. This right here is the 96 well plate that we use where we combine everything into the well and then run through the microplate reader. And so just knowing this layout, if you picture that later on, it'll be helpful when I'm going through like how we set up our experiments. So we use that in the microplate reader, and we perform fluorescence assay on AFB1 concentrations and absorbance assay for erythropolis cell concentrations. So our project 1, our goal, we wanted to begin by determining wild-type bacteria strains performance when it is combined with the toxin. So the first thing we needed to figure out was whether or not there is an antibiotic effect on the bacteria caused by the toxin. This was extremely important because it suggests that there is a burden placed on the bacteria's fitness, and as the toxin begins to have a stronger antibiotic effect, selection for degradation becomes more powerful. So in order to test this, we ran an analysis of toxin fluorescence, erythropolis fluorescence, and optical density. And we set up, so the 96 well plate that I pointed out to you earlier, this is just how we set up and visualize it. So each of these squares represents a well in there. And so we use low concentrations of bacteria and toxin concentrations in increments of 2 micrograms. So you can see we have 3 replicates of each toxin concentration. And we knew from previous experiments, we assumed the antibiotic effect takes into effect between 10 and 50 micrograms. So that's why our plate was run from 12 to 48 micrograms, as you can see. This is the data from our plate on bacterial fluorescence. So looking at this, we can see that these 3 right here are the 18 microgram per milliliter concentrations, and then the next is 20 micrograms. So we can see between 18 and 20, there's a very drastic change in the bacterial growth. So we believe that this is the threshold between which the antibiotic effect really begins to take effect. But you can see that there is still growth here, so that is important for us. So knowing this, we attempted several rounds of artificial selection to improve the degradation dynamics. We believe that if we could push the boundaries of the antibiotic effect, we can increase the power of selection due to the pressure placed on the bacteria. We already looked at how the toxin affects the bacteria. So for the second part of our experiment, we focus on how Rhodococcus erythropolis degrades AFB1. So our experiment starts, we inoculate our bacteria, 5 different strains, in cultures in buffered media and 15 micrograms of toxin for 72 hours. After our bacteria grow up, we set up our plate like this. So we have 5 replicates of each strain of bacteria, both in unbuffered and buffered media. So the plate is run for 48 hours, and then we can analyze our data. So we look mainly at the toxin degradation curves in this one from the fluorescence, and we're really focused on the slope of the degradation because that tells us about the rate of degradation. So based on that, we select our 5 best degraders, and we remove those from the well, and they're inoculated again, and the process is repeated. So we perform this for approximately 5 rounds. Looking at our data, we did not notice any difference between the degradation curves of conditioned bacteria and the wild type, which is what we expected to see. So unfortunately, that's not what we saw. But we did see there is a difference between the buffered and unbuffered media, where the bacteria appear to degrade better in unbuffered media. So because we want to select for more efficient degradation, we had to change our selection scheme as it wasn't working. So there are many different variables that we could have altered. Any of these could be altered. In particular, we wanted to put pressure on the bacteria, so we decided to test changing the toxin and bacterial concentrations. From previous experiments, we knew that the bacteria begin degrading around 0.5 OD, or close to their carrying capacity. So you can see in our high bacteria rows that they grow very rapidly, and you can see that the toxin degradation begins much sooner in these than in the ones with low bacterial concentrations. So this was really awesome for us to know, because one of our goals is to degrade the toxin as fast as possible. So if we use a high bacterial concentration, the toxin will be degraded much sooner. In addition, we looked at high toxin concentrations. We believe that at 25 micrograms, which is what we used here, it is high enough that the selection for degradation must be taken into account by the bacteria, but it's not yet deadly. So this was important for us for looking for a specific phenotype. It makes sense that it would occur at a high concentration like this. And so we noticed something interesting in the curves we just saw. So if you look here at this well, which is high toxin and low bacteria, a normal as in like typically what we've been seeing in our wells is that the toxin degradation occurs approximately as the bacteria reaches its highest points, as it stops growing. And in this well, we see that the toxin degradation occurs as the bacteria is beginning its exponential growth. So this was very interesting data that we saw. It was the first time we've really noticed this. This suggests that the degradation is a result of secondary metabolism. So the bacteria are going through their primary processes, and degradation is just happening as a side effect of those and not necessarily the target. This would suggest that if it's happening as the bacteria are growing, that the degradation for these bacteria were more intertwined with the primary processes and may be critical for the toxin to be degraded in order for the cells to grow. So we have since been trying to repeat these results and use this information moving forward with our current tests. So some of the things I just hope that you take away from today. So we focus on aflatoxin B1 because it is a prominent and harmful mycotoxin. It is the most toxic naturally occurring carcinogen known, and it is the cause of over $1 billion lost annually. Additionally, AFB1 has an antibiotic effect on Rhodococcus erythropolis, which causes selection pressure and allows us to possibly artificially select for better degraders. And using this, we found 2 different degradation trends through our experiments, one where the toxins are degraded after exponential growth and one where they're degraded before. So moving forward, we really want to determine the mechanism for toxin degradation. We want to determine if the enzymes used in degradation are an oxidase by treating the culture with an antioxidant, and hopefully from this information we can identify the enzymes involved in degradation. Additionally, we would like to one day perform droplet-based microfluidics to isolate cells containing low-frequency traits so that we can actually see the traits that are not overshadowed by the larger population. So I just want to thank a few people for this summer. Professor Momeni, who is not here, for letting me come into his lab and all his self-help this summer. Marco for teaching me everything and letting me mess up all of his experiments many, many times. Sales, who was my partner through this experiment. Ken Birch and Laura Ann Lowry for setting up this REU and letting us all come here. And for the entire Momeni Lab team, and Matt, just for being the kindest and most welcoming lab and for answering my millions of questions. So thank you guys for listening. Anyone have any questions?
So on the charts you were showing us with those lines representing like each trial, were those lines of the concentration over time or the rate of like degradation?
So they're the fluorescence of it over time, which is associated with the concentration over time.
Okay.
What are some other ways that these toxins can be removed from like foods and stuff?
So there are some physical and chemical ways. So for instance, like chemicals, they can use ozone and ammonia to destroy the toxins, but they don't prefer these because they're not applicable to all mycotoxins, and they can be harmful to people. And they use physical methods. They use absorbents such as clay and charcoal, and they add them to mycotoxin contaminated diets, and they're really trying to actually target the gastrointestinal tract there. So it's not something that removes toxins from food, and so that's why we focus on bioremediators for this instead of those.
You mentioned at one point, I think you called them wild type bacteria. What are those, and how do you select them?
So the wild type are the non-selected bacteria. So those are just straight from like the original colonies. They've never been in the presence of the toxin, whereas we compare those. So those are our control, and we compare those to the conditioned bacteria that have been repeatedly exposed to the toxin.
Oh, okay, got it. So at some point they're also exposed to the toxin, and you look at the difference.
Yes.
Okay, got it.
Why did you choose to focus on the toxin that you did? compared to others?
So AFB1 is one of the most dangerous toxins, mainly because when it's metabolized, it's broken down into a slightly different structure, and that structure causes gene missense mutations at some tumor suppressor genes, and so you get cancer as a result of that. And so AFB1 is particularly very dangerous in that sense.
So if eventually you were able to do this successfully, where would you apply it? Like in storage where food would be?
Yeah, so I believe the goal would be that you would, after, so the fungi grows while it's in storage typically, or in transport or something. So before, you bring food out to market or give it to animals as feed, you would want to expose the bacteria, hopefully, that we use to the toxin to degrade it. Okay, well, thank you, everyone.